Giardia is an organism common to most areas of the world. Giardia can present as either acute or chronic diarrhea, which may wax and wane over time. Animals with Giardia may also have weight loss, poor weight gain, or anorexia. Let's look at the life cycle of this organism. A recently ingested Giardia cyst is being digested in the host animal's small intestine. From the cyst, the trophozoite form is released and begins colonizing in the small intestinal lumen. The trophozoite then adheres to the intestinal wall and begins interfering with small intestinal mucosal absorption. Trophozoites are released from the intestinal wall and begin to move down the intestinal tract, where the surrounding environment begins to change. The intestinal contents are less fluid, pH increases, and cholesterol levels drop. The trophozoite senses these changes in its imminent release into the outside environment. The trophozoite will not survive long outside the host, so it begins its life-saving morphologic change to its cystic form. Encystation-specific vesicles begin to form within the trophozoite and migrate to the surface. From these vesicles, a cyst wall protein is excreted that eventually forms the Giardia cyst. Unlike the trophozoite, the cyst is very stable outside the host, especially in cool, moist environments. The life cycle repeats when an unsuspecting host ingests the Giardia cyst. Traditionally, in-clinic diagnostics have relied on Giardia cyst detection on a fecal flotation, but because cysts are intermittently shed and very small and fragile, the accuracy of microscopic methods is usually poor. Identification of trophozoites is also utilized, but this form of Giardia is not generally present outside the host. During the encystation phase, when cyst wall protein, CWP, is being excreted onto the surface of the trophozoite, a good amount of the protein escapes into the surrounding fecal material. It is this protein that the IDEX SNAP Giardia test identifies. Let's look at how the new IDEX test detects Giardia. The user swabs the fecal sample with a convenient swab, and the swab sample is then mixed with a conjugate. The conjugate contains anti-Giardia antibodies that have a specific affinity for the CWP and therefore bind any CWP present. These antibodies are also tagged with a molecule, represented here in blue, that will assist in antigen detection later in the process. The conjugated sample is then pipetted onto the SNAP test with the swab, and the test is snapped after the sample flows up the matrix. Let's look inside the test to see what happens. The sample is pipetted onto the test and flows up the matrix. Bound to the matrix are more anti-Giardia antibodies that will bind any Giardia antigen that is floating by. The Giardia antigen is captured between the antibody bound to the matrix and the antibody that has the blue tag. The user then snaps the test, which initiates the flow of wash solution. The wash solution cleans away any non-bound material, which clears the read window. The wash solution is followed by a substrate that, when interacting with the tag molecule, generates and amplifies a blue color. This heightens the ability to detect positive animals. Unlike unreliable in-clinic microscopic methods that rely on cyst detection, the reliable ELISA platform of the SNAP Giardia test can greatly improve your accuracy of Giardia diagnosis through target antigens that are more evenly distributed in the fecal sample.